Let me take this opportunity to share with you some example illustrating the impact of design-driven biotech in the field of personalized and precision clinical neurology. New targeted therapies for autoimmune and inflammatory diseases would require greater understanding of a patient or a person at risk to get the therapy personalized for either of those subsets. In this term, biomarkers enable pre-early subclinical diagnosis, guide targeted therapy, and monitor the activity and therapeutic responses in many diseases. The above mentioned is so much valuable and important since chronic autoimmune inflammation causes any kinds of autoimmune inflammation causes is structured to consist from different stages, including subclinical and clinical one, multi-step process. Multiple sclerosis, MS, is just one of the chronic tissue-specific autoimmune disorders resulting in a destruction of myelin by different tools, including autoantibodies. The crucial step in the pathogenesis of MS is a primary myelin damage, which is mediated by autoantibodies to trigger a release of separate and pathogenically valuable myelin-associated epitopes into the bloodstream. Those epitopes act as a sensitizing factors to generate autoantibodies more and more, which in turn would drive the demyelination and thus the disease progression. Anti-myelin autoantibodies, about 20-25 today known. Different cytotoxic mechanisms, antibody-mediated damage, and bloodstream. At present, a spectrum of those antibodies confirmed to be very large. The crucial autoantibodies are depicted here. The main one, anti-MBP, myelin basic protein. Along with canonical antibodies, as I've already mentioned, some of the families proven to occur are antibodies possessing with catalytic activity. Today, we would not concern other ones except proteolytic. Enzymes, AB, antibody, zymes, enzymes, and thus to belong with antibodies with the feature of functionality. Meanwhile, the likelihood of autoantibodies and biochemical evidence for MS has been proven to be proportional to the presence of anti-myelin antibodies and NBP in particular. Look. It's just the introductory. The examples of antibody-proteases are IgG found in a series of autoimmune disorders, including autoimmune myocarditis, autoimmune thyroiditis, and lots of others. It's not so crucial today. A situation of phenomenal interest is occurred in MS, multiple sclerosis, which is a chronic autoimmune inflammation in CNS, central nervous system, restricted by a specialized tissue and resulting in demyelination, axon loss, and development of disability. The latter mentioned would strongly demand a stage of remyelination, including regenerative and rehabilitative manipulation, where anti-action of antibodies in terms of engineering are strongly needed. Just later. We have established that anti-MBP antibodies harvested, collected from MS patients and mice with MS analog, experimental autoimmune encephalomyelitis, has exhibited MBP-specific proteolytic activity of the MBP molecule. Only MBP, not other myelin-specific antigens. The activity markedly differs between MS patients and healthy controls. And also in patients with different courses of MS. Look, very aggressive courses. Exacerbation in remittent in red. Progression in progression in red as compared with white. And finally, the activity demonstrated significant correlations with scales of demyelination, neurological deficiency, and thus with the disability of the patients. You see it from the ADSS Husky score. Of the greater interest is a sequence specificity of antibody proteases, which was analyzed by screening peptide cleavage products by a combination lots of the latest innovative technologies. Just look, including mass spectrometry, gas chromatography, and lots of. As you might see, antibody-mediated proteolysis of MBP results in generating set of peptides with molecular masses ranged in various but fixed boundaries to suit common principles of the molecular architectonics of MBP. The final statistic data being based on AI, artificial intellect, revealed 5 sites of preferential catalysis within the molecule of MBP. Most of those sites are located within 43170 flanks of peptide. The sites from the set located within that range sequence would comprise 81103 and 8298 subsequences that, while being sequence specific, highly immunogenic, very specific and major inducers of very aggressive MS analog in mice. The extra 2 sites from the same 43170 set are located within subsequences that, whilst being sequence specific but low immunogenic and encephalopatogenic, both prove to be inducers of moderate MS analog. As we have established, the most immunogenic and encephalopatogenic sequences of immunodominant category responsible for generating aggressive bursts of demyelination are concentrated in 3 principal areas of MBP molecule. The strongest one is in the smallest A298 subsequence. A weaker is formed by 81103. The final step, along with the sequence typing of the studies, concern the evolution of antibody-associated proteolytic activity at different stages of the disease progression. For these purposes, we have been monitoring MS patients, their direct relatives, and healthy controls. Look. (70% and 20%, respectively, of the MS patients and MS relatives were initially seropositive for canonical anti-MBP antibodies with no proteolytic activity, so-called disarmed antibodies. And less than 2% of the MS relatives were initially positive for antibody-proteases, but with very low activity, low active. During the evolution, 0, 1 year, 2 years, 3 years, MS relatives being initially seropositive for disarmed antibodies and very low active antibody proteases demonstrated stable growth of antibody proteases. Being past approximately clinical, subclinical, and clinical stages. Okay. Oh, sorry, 5 seconds? Minutes, it's okay. It's much more than seconds. Yeah. No detail more. Along with changes of the sequence specificity, when we saw an extensive growth of the activity, we could predict transformations between the clinical courses, progression, progredient, and others. Changing of a remittent type as moderate one into secondary progredient type, which would dictate changing of treatment. The translational potential of this knowledge has been suggested us by 3 American biotech and biopharma companies. And it's in the rational design of new diagnostic tools and new therapeutics. In this sense, antibody proteases, along with canonical antibodies, but canonical antibodies are able only to bind. Antibody proteases, whilst possessing functionality, are able to catalyze. And those can be programmed and reprogrammed. Of tremendous value in this sense are antibody proteases directly affecting the physiological remodeling of tissues with multi-level architectonics. You clearly understand that's, for instance, myelin. So further studies on antibody-mediated MBP degradation and other targeted antibody-mediated proteolysis may provide biomarkers of the next step generation, or even principally new biomarkers family. So the companies from 3, we already signed 5 years agreement. We are basic researchers and considered to be clinical researchers. American company is design-driven bioengineering and translational applications. Just briefly, what the next step of the manipulations with the data accumulated. Thank you so much for your patience.